Thanks very much, Marie. And Gwen, thanks for the introduction. Here we're all here together to discuss measuring sweetness, and it's a pleasure to address this after thinking about it for several years. The committee, through a series of discussions, we landed, the committee to which I'm an advisor, landed on, very good. So we've been discussing this on the committee for a number of months, and we landed on this topic of sweetness as a discrete and tangible way to take these discussions forward and develop the needed knowledge that is a gap. So first, I'd like to acknowledge some of the many people who've been involved in this. Many of them are here. I'd also like to acknowledge my co-chair, Dave Klurfeld, Marie Latulippe, who's done a wonderful job, and Char Myers, who I think all of you know because of all of the arrangements she's been doing. So we've got a diverse audience here today of all sorts of people, and some of us are not sensory scientists, and so I thought we could start, at least for a moment or two, to set the meeting up with why we're here and also why we're focusing on sweetness today. Now, it's important to keep in mind that sweetness doesn't necessarily equate to sugars, and that was the first thing that I think is an important point to make. But sugars certainly have a role in the thought process that brings us to sweetness. And so this diagram is one way of thinking about it. If we look at what we think we know, and we do know most of these things, we know intake of added sugars is excessive, that sweets may be high in added sugars, that excess added sugars or excess sweet taste contributes to excess calorie intake, and that excess caloric intake leads to increased risk of obesity and related non-communicable diseases. Whether the arrows are causative is another question. But basically, if we look now at several global authoritative health organizations, they're suggesting that sweet foods should be avoided or limited. And let me just show you a couple of them. If we look first at the first three, at the very top of the slide, the ones in North America, since this is ILSI North America, you see the Canadian statement, the American Diabetes Association, and the American Academy of Pediatrics statements, all suggesting different things about this sweetness that need to be researched. If we look at the bottom four or three, internationally, there's also a great deal of interest and concern. The reality, though, is that if you read the reports from which these snippets are taken from, the evidence for some of them is not as strong as perhaps we would like to see, and it's certainly something that merits more discussion and thought. So why are we here in terms of where others have been in the near past? The first was a meeting that some of us attended and others did not at NIH. It was held just about a month ago on sensory nutrition and disease, and it covered a whole range of topics to understand the hedonic pathways linking taste and sensing of food, and also understanding the physiological roles of chemosensory receptors and how these taste and smell sensors are involved in nutrition. There were a few mentions of sugar. I think, Gary, you spoke at the meeting, and there were a couple of others where they got into this particular issue, but there were many other talks on other things. So the meeting was fairly heavily focused on physiological mechanisms, and most of the evidence presented, or much of it, was on animals and not necessarily on humans. And it was evident also that there was very little known about some of the interactions we'll be talking about today in humans. So that's one recent meeting. And another was just 2 or 3 weeks ago in Dusseldorf, which was the Codex Committee on Nutrition and Foods for Special Dietary Uses. And I understand from colleagues who were there that the meetings went 14 hours and more every day. And they got, I guess they did feed them, but that was about all. The discussion that was most relevant there, from what we hear, was about ensuring that carbohydrates in follow-on formulas that are non-milk protein-based didn't contribute excessive sweetness. And the question is, how do you determine what excessive sweetness is, and how do you measure that? So those are two sort of recent meetings that certainly focus on some of the things we're talking about today. I found it very useful in talking with several of you who are experts in sensory science about why we need evidence for the role of sweetness in the diet, to have you help me by contrasting the issue of salt and sodium, salty taste, and sweet taste. And this cartoon summarizes it, and I think it's been Gary who's been so helpful in putting some of these down on paper. So we have the taste, the related ingredient, the method to assess this in diets, the biological drive, accepted intake biomarkers, and related ingredients and whether they have effects on health outcomes, and then can you measure preference? And you see for salt and salty taste, pretty much all the boxes are filled, and there is health guidance about this that's adequate to give to the public. When we come to sweet, there's some gaps here, and we have ingredients that include sugars and non-caloric sweeteners and proteins. The method to assess them in the diet is absent or under dispute, I guess we'd have to say. The replacements are available, at least some of the replacements. Biological drive, yes, but whether there's an accepted intake biomarker, not clear. And whether there's a short-term preference change, perhaps yes, but long-term, we don't know. So there's still a lot of questions there. Also, another way to look at it is to think of a hypothetical pathway from perception or intensity, which is what we're here to study today, to health. And to start out by considering something that's really not so value-laden as some of the things we'll be talking about today. Think of orangeness and the perception analysis, and then the hypotheses and health implications that come from this. And first of all, the perception, the taste of limonene in oranges, but also in lemons and some other of the citrus fruits, is the taste we're talking about here, or the odor, whatever. The quality of the smell or the flavor of orangeness is pleasant, at least to me. I love that citrus taste or smell. Then there are hypotheses that we might construct about them. One would be that exposure to orangeness influences the liking for orangeness and for oranges. and that liking for orangeness results in eating more oranges, and eating more oranges is good, and the health outcome is good. Now, if you'll bear with me for a minute, maybe we can do the same thing with sweetness. So if we think of the perception, the taste of sugars or low-calorie sweeteners, the quality of the taste or sweetness is pleasant, at least to most people. One hypothesis might be that exposure to sweetness influences the liking for sweetness and for sweet substances. And that hypothesis of liking for sweetness results in eating more sweet foods. And the more sweet foods that are consumed, the worse the health outcome is going to be. Okay, so that's what we think we know. And then there's a whole bunch of things, however, that we probably don't know. What's the role of sweetness in terms of, for example, is sweet in the diet the same as sugar in the diet? Does sweetness intensity in the diet cause a preference for sweet, promote preference for sweet, cause adverse health outcomes? And how should sweetness be defined to address these questions, which I think are all relevant? We have to have a definition, and we also have to find some ways that we can measure sweetness in foods and in beverages and diets. So we hope by defining and clarifying the method of measuring sweetness that it'll allow for, first of all, harmonization across studies and meta-analyses, because at this point it's a lot like studies of some other things where everybody defines the term differently, and so it's very difficult to do a meta-analysis and put them all together. So that's the first thing. The harmonization can help in interpreting the data that exist. And secondly, and downstream, perhaps acceptance of the data by authorities that develop dietary recommendations globally would make them more understandable. So today's focus is on sweetness intensity. Sweetness intensity, as I understand it, and again, those of you who are sensory scientists can object as soon as I finish, which will be very shortly. It's more binary compared to preference. So it's a yes-no sort of thing. It's more objective. The differences among individuals are relatively small compared to preferences. And it's a lot easier to start with developing measurements for it than is the case for other things. So what's out of scope today, just because we only have 6 or 7 hours, is sweetness preference, which is much more subjective. It is liking, it's an hedonic quality, highly variable by individual, and certainly more difficult to translate and generalize. So for today, we're focusing on sweetness intensity. Now, how do we test hypotheses to address these questions? Well, today's goal is to try to figure out ways of measuring dietary sweetness. But the other ways we can test these hypotheses, of course, are experiments and varying sweetness experimentally. And there's several studies underway, which perhaps the sensory scientists are well familiar with, but some of the rest of us, at least I speak for myself, I wasn't aware of. And so I'm asking Dave Klurfeld to briefly review 2 studies that are relevant to the sweetness pathway that we've been talking about. These studies are, in fact, limited in that we don't really know and have agreement upon means for measuring sweetness. But the studies will now be described, at least very briefly, by my co-chair, Dr. Dave Klurfeld, who's a national program leader for USDA's Agricultural Research Service and an old and dear colleague and friend. David has a long history in nutrition that most of you probably know about in terms of experimental nutrition, and we're very lucky to have him here in Washington now, working also on the policy side of things. David?
Well, thank you, Johanna, for that introduction. There are two groups currently funded to address some of the questions Dr. Dwyer was just talking about. And we're going the wrong way. Okay, now we're doing it correctly. So Gary Beecham, in collaboration with some of my scientists, David Baer and Janet Novotny, at the USDA's Beltsville Human Nutrition Research Center, are funded from NIH through a cooperative agreement to determine how reducing sugar in the diet affects perceived sweetness. And this is an attempt to get at sweetness of the entire diet rather than specific foods, which, to my knowledge at least, there's no existing data on. And the attempt will be to see how reduced sugar affects the most preferred concentrations of sugar in both foods and beverages. And so the design is to have four groups. The control group consuming about 150 grams of sugar a day, and about 3/4 of that will be through added sugars. Then a reduced sugar group that gets a reduction all at once. The third group would be a gradual stepwise reduction in 3 steps to get to the maximal reduction over 3 months. And then a low sugar group that has the same intensity of sugar but supplied through non-nutritive sweeteners. And the hypotheses to be tested in this are that the perception of sweetness along with reduced sugar intake will increase, and the preferred sugar concentration will decrease during that time. To me, what's really interesting is the NIH institute that's funding this is communication disorders, not one of the more metabolically oriented institutes. And I think that's a reflection of the part of the problem of the science in this whole area, that the traditional health-associated science groups don't seem to care about this as an important issue, although it's critically important in making health recommendations. There are a number of secondary analyses that are planned. The required poop collection in any modern study to look at the fecal microbiome, a number of blood samples to look at metabolic hormones, not just glucose metabolism, but a variety of other factors that are thought to drive the desire for eating, and particularly for sweet. A number of behavioral questionnaires about food cravings, food satisfaction in relationship to changes in sweetener intake, and then looking at taste receptors. In addition, we have very fortunate to have brought Dr. Kees de Graaff over from the Netherlands. He's going to be speaking later this afternoon, and he's doing a similar type of study but with a different experimental design. And he's pointed out that the statement that less sweetness exposure induces a lower sweetness preference, leading to lower sugar and energy intake, which could result in lower body weight, is simple, attractive, and powerful, but not based on very much objective data. And so the objective of Dr. de Graaff's study is to do a slightly longer 6-month intervention, looking at both lower and higher levels of sweetness compared to the standard Dutch diet, and look at changes in perception and preference for sweetness and saltiness. This, rather than a controlled diet from our metabolic kitchen in USDA, will offer ad libitum choices to the volunteers in Dr. Graaff's study, look at glucose metabolism, body weight, and compliance measures. So the outcomes that will be tested are changes in perceived intensity and liking of sweetness for both sweet and savory foods, familiar and new foods, through a variety of measures of psychohedonics and psychophysical functions. Changes in food choice and taste patterns and energy intake will be measured, as well as a similar list to the USDA NIH-funded study on quality of life and well-being and appetite and body weight, et cetera. So the design of Dr. Graaff's study looks pretty much like this over 6 months. The first group exposed to their usual intake of sweetness. Another group with a reduced sweetness diet, and one with an increased sweetness diet over 6 months. So today's objectives are to describe the challenges and opportunities for measuring intensity of sweetness, to look at key considerations for measuring sweetness, to outline new potential research questions relevant to both the study designs I've described and health endpoints, and identify next steps to get everybody on the same page, measuring sweetness measurements. So again, to reiterate what Dr. Dwyer said, in scope, we're interested, very focused here on sweetness measurement, what questions we need to address. We'll have Dr. Paula Trumbo talking about the policy and regulatory situation related to sweetness, and we'll have other folks reviewing the methods to measure sweetness intensity in both foods, beverages, and total diets. And I think that latter aspect of total diet is the real challenge here. And again, re-emphasizing that addressing the role of sweetness in diet and health is not what we want to get diverted with. You know, that could take multiple days by itself and still not come to any agreement. And we're not going to, even though we're going to get some background on policy and regulation, we're not going to discuss that. So just to tell you how the day will progress, we're going to have half a dozen speakers. We're going to have limited time for questions and answers after presentation, but we're also asking you to write down any questions and submit them to us, because at the end of the day, when you folks get to go home, the speakers and the steering committee are going upstairs to hash out some of the unresolved questions and try to actually prepare a manuscript from the last 3 presentations of the day. So with that, we're going to move into the scheduled talks, and our first speaker is Dr. Nadja Berenstein. She's a science and technology historian and journalist who writes about the history and future of food. Her writing has appeared in Forbes, The New Food Economy, Food and Wine, Serious Eats, among others. She's won a 2019 James Beard Award. She hasn't offered to cook for me yet, but that...
For writing, not for cooking.
Oh, that's right. Her award was for reporting on the history, technology, and ironies of clean label description, which is a fascinating area. Dr. Berenstein has been a featured guest on NPR's Science Friday, multiple podcasts. She was an advisor for the Museum of Food and Drink's inaugural exhibition, Flavor, Making It and Faking It. You can read more about her research about flavor and food history on her blog called Flavor Added. Please welcome Dr. Berenstein.